right, so you guys will get all the questions today. That's a good thing, right?
Yes.
So today we are going to be talking about cartilage and bone. You guys basically can tell me everything about this presentation so far, because cartilage and bone gladly are specialized form of connective tissue. So seeing that they're specialized forms of connective tissue, what would you expect cartilage and bone to have or be comprised of? Collagen, yes. What else? It's a specialized form of connective tissue, so it should be like any connective tissue. You guys did very well yesterday, by the way, on the connective tissue tutorial.
The ground substance in this case wouldn't it be like a hardened, become hardened?
Yes, that's good. So the ground substance would contain a little bit more to it. If you look at cartilage versus bone, what would you expect the consistency of a cartilage versus bone to be?
Cartilage would be softer than the bone.
Exactly. So cartilage would be semi-solid, bone would be more solid.
Yeah.
So cartilage and bone are specialized connective tissue, and they are derived from embryonic mesenchyme. You already know that connective tissue contains cells, extracellular matrix, and you know what extracellular matrix is. There are fibers. What are the fibers again you can find in extracellular matrix?
Collagen fibers, reticular fibers, and
Elastic fibers. Exactly. We actually covered some cells that you could find in cartilage and bone. You remember any cells? What type of cells?
Chondrocytes.
Chondrocytes. Exactly. So you have most right already. Bone, what would you expect to find in bone? What type of cells? We did speak about them briefly.
Would plasma be there?
Well, yes, in the bone marrow. So in terms of the cells that would make up the bone itself, you'd expect osteoblast, osteoclast. Well, there are different types of cells in the bone that is responsible for making the bone itself, mineralizing the bone, making the hard consistency of the bone. Osteoblast, we spoke about osteoblast briefly in one of our lectures, not so much in detail. And remember we said that the blast cells are the immature version of the cell. You already know that chondroblast or chondrocytes, you find them in cartilage, and you should already know now that you find osteoblast, osteocyte, and osteoclast in bones. This diagram is just trying to orient you a little bit on the structure of the bone. You have different types of bone in the body. You have long bone, you have flat bones, for example, in the skull, long bones in the axial skeleton, making up your limbs, the femur, this long bone, the longest bone in your body, this bone in your leg, the femur. But in terms of the anatomy of the bone itself, the bone is made up of articular cartilage, and at the end of the bone, we call this the epiphysis or epiphysis. Also have an area in the middle region you call the diaphysis, metaphysis. What's important about this is that there's also cartilage on it. So we're talking about bone and we're talking about cartilage. There are different types of cartilage. I can already tell you everything. Hyaline, elastic, fibrocartilage. Did you guys do any reading on this topic?
Not really, but you really did touch on bone tissue in, I think, was the human physiology class.
Yeah. Do you know about cartilage, the different types of cartilage that you have in your body?
Can we learn it? Yes. If I can remember everything.
All right. I'm trying to elicit memory, but if you can't remember it, it's fine. That's why I have in this session now. What's important about this diagram, though, is that there are different areas of the bone and there's special function based on the area. The bone can also be divided into spongy, which is usually in the middle part. They tend to call this the trabecular bone, and it can also be divided into classified as compact. So it can be classified as spongy bone, compact bone. Compact bone is the hardened area that is usually on the external surface, and the most external surface of the bone, you call it a periosteum. Here we have the epiphyseal line. The epiphyseal line is important because this area is an area on the bone where if this area undergo calcification, because you know the bone is hardened because it's calcified, right? Ossification, calcification. If this area is calcified or undergoes ossification, then you basically stop growing. This area on the bone is the only area that is is not really too hard or undergoes ossification because cell division has to take place in different ways in order for the bone to grow. And here we have the cartilage. There are different types of cartilage. As I said, you have fibrocartilage. Fibrocartilage you can find in, for example, intervertebral disc. Hyaline cartilage you can find here, like articular cartilage. Here you would expect to be hyaline cartilage. There's difference between hyaline fibrocartilage and elastic cartilage, and we'll get to look at that. What's the difference between bone and cartilage? We know that cartilage is semi-solid, bone tend to be more solid. They both contain cells and extracellular matrix, but they contain different cells. We already know that for the cartilage you have chondrocyte, for the bone you have osteoblast, osteocytes. When we talk about chondrocytes, we mean that the chondrocytes are responsible for making the extracellular matrix. The chondrocytes are the cells in cartilage that are responsible for making the extracellular matrix, while the osteoblasts are the cells in bone that are responsible for making the extracellular matrix. And you guys already know what the extracellular matrix is. Consists of fibers. You have different types of fibers. Also consist of brown substance. You guys also already know that there are proteins, complex protein structures, as well as minerals for calcification. There are 206 bones in your body, and just like the cartilage, the bone has a matrix. The bone also has fibers, collagen, ground substance. The difference though is that the bone will contain mineral, and this mineral, calcium hydroxyapatite, it's responsible for hardening the bone. The difference though with bone and cartilage is that the bone is highly vascularized. It gets 10% of the cardiac output. So 10% of the blood that leaves the heart goes to the bone. Cartilage does not really have much blood supply. Problem with cartilage is when you damage the cartilage, the damage tend to be permanent. While if you fracture your limb, you fracture anywhere on your body, depending on the area that you fracture, if that area has a rich blood supply, the blood will just regenerate. The osteoblast will just come and regenerate, recreate the collagen, recreate the extracellular matrix, and you get a freshly formed bone. And that's a good thing. We can divide the bone into spongy based on the location and the physical characteristic. Can be spongy or it can be compact. Compact bone accounts for 80% of the weight of the bone. The spongy, 20%. When you look at the periosteal region here, I showed you in the first diagram, periosteal region, where you expect the compact bone to be, what you have is what we call osteon. These little round things. What they are, very simple. They're osteoblasts. Remember, we said that the osteoblast is responsible for synthesizing the collagen as well as the extracellular matrix. Basically, what will happen, you have an osteoblast that creates collagen or spill collagen, and this would be the osteon. So the osteon is the osteoblast creating collagen, and if the osteoblast is creating collagen, what mechanism of release would you expect that collagen to be?
Exocytosis.
Exocytosis, exactly, because it's leaving the cell, right? So the osteoblast creates the collagen and it's basically just spit it out by exocytosis, and that's basically the osteoid. That's the organic part of the bone. Here, I think this is just showing the fiber patterns. This is just showing fiber patterns, and this would be collagen fibers. This is showing the collagen fibrils down here. And as I said, the organic part of the bone is the osteoid. So it's the osteoblast just spitting out the collagen. When the bone is mineralized, however, that's the inorganic part of the bone, and you'll get to understand that a bit more. So this is just showing collagen amino acid. All right, so this is a better look at the bone itself in terms of the structure. You have the osteon. The A version system is the bone with the collagen fibers, and you usually have blood vessels in it, and that's basically basically the aversion system. So you have the blood vessels, nerve channels within this osteon, and you call it the aversion system. Here, you're seeing this as osteocyte. Remember that we said that the osteoblast will produce the osteoid. Well, if the osteoblast produces the osteoid, when the osteoblast matures, what will it become?
Osteocyte.
Becomes osteocyte. Exactly. Very good. All right. This is showing the periosteal layer. Periosteum is a dense connective tissue. So that's just the external layer of the bone itself. And here would be the compact region of the bone, the hardened region of the bone. The hardened region of the bone, you expect to have the osteoid. So you have a lot of collagen, and you tend to have osteoblast transform into osteocyte. When the osteon, which is this area here, and if you look here, you have multiple of them, right? Multiple osteon. It's just one of the osteoblasts. For example, an osteoblast just comes, spill out the collagen, and eventually it becomes circumferential. So sometimes you see, or here they call it circumferential lamellae. So
yes.
here,
I was agreeing with you.
Oh, okay. Yes, here we have spongy part of the bone. So we said that here is the compact part of the bone, spongy part of the bone. You can already take note, it's right here. You can already take note of, well, Sharpie's fibers is just collagen fibers that is sticking in. So you already know that it's just the osteoblast that produce collagen fibers. Sharpie's fibers are just the collagen that are sticking in. And you can also take note of what is called aversion system. I think I saw that, the aversion canal, right? So this is the canal in which you have blood vessels running, and this is also another way in which there's communication within the bone. Because imagine, it's very hard, it's very tough. So you'll have to have channels or canals just running within it in order to supply blood from the toughest area, which is the compact area, to the soft area, which are spongy area, which would be the medullary cavity. We look at the bone matrix. We said that the bone matrix was a bit harder than the cartilage matrix. Cartilage matrix is more gel-like, but it's firm, right? You expect water, you expect collagen, you expect proteoglycan. The proteoglycan is the bigger term for the protein that exists with sulfated group, and where the glycose aminoglycan is attaching to the core component of the proteoglycan. We already said that cartilage matrix is avascular. There's no nerve supply, there's no lymphatic supply. When you have a damaged cartilage, you tend to not have a regeneration, while the opposite is true for bones. If the area that is damaged is highly vascularized, then you can have a regeneration of the bone itself. So the components of the cartilage, we already know, consist of the cells. We know they are the chondroblast, the chondrocytes. Sometimes you might say fibroblast in the cartilage as well, because sometimes the fibroblast can convert or develop into chondroblast or chondrocyte. The ground substance you expect to be the glycosaminoglycan, hyaluronic acid, chondroitin sulfate. The bigger term is proteoglycan. And you also expect collagen fibers as well as elastic fibers in cartilage. So this is a better look at the proteoglycan structure. This is the core protein, and as I said, it's basically chains of sulfated groups. So you can have heparan sulfate, chondroitin sulfate. So they're just different sulfated groups that are anchored to the core protein. And of course, you expect glycosaminoglycan as well. The difference, however, with cartilage and bone is that the collagen type can be different. So sometimes you might see bone, you tend to have type 1 collagen, while cartilage, you tend to have type 2 collagen. And you know that the collagen can be different in terms of the structural composition based on the protein. This is just showing cartilage, what you expect to find. So what does the cartilage look like? You can have fibroblasts in the cartilage. Here you you have a perichondral fibroblast, and at the periphery you tend to have the chondroblast, right? So this is the perichondrium right here. So you'll find that the outermost layer of cartilage tend to be called perichondrium. Not all cartilage, because remember that you have different types of cartilage now, fibrocartilage, elastic, you have different types. Some you might find won't have the perichondrium, which is the outermost layer, but if you look in the outermost layer, you tend to see the fibroblast as well as the chondroblast, and you also expect to have your fibers running between the cells, because remember that the chondroblasts are going to synthesize the extracellular matrix. In this case, it's cartilage, so it's chondroblast, chondrocytes. Expect them to synthesize the extracellular matrix. But when you look a bit more in the interior of the cell, you see the more mature form of the chondroblast, which is the chondrocyte. Territorial matrix just means the matrix that is close to the cells or closely approximated to the chondrocyte, because it synthesizes the collagen and naturally spit it out by exocytosis. So sometimes you'll have some of the collagen fibers that are closely approximated to it or within the territory. And then you also have interterritorial matrix where it's between the cells. You should expect a lot of rough endoplasmic reticulum in chondrocytes, right? Should you?
Yes, since a lot of protein synthesis taking place.
Exactly, and you can apply that knowledge to all the other cells that we'll look at. For example, the osteoblast, if it's synthesizing so many things, synthesizing the extracellular matrix, it has to have a rich, rough endoplasmic reticulum composition. This is a more confusing look at everything that we've discussed so far, but let's go through it slowly, because understanding this, we can go through the lecture very quickly after that. So we talk about cartilage. You have different types of cartilage. Hyaline cartilage you tend to find on the articular cartilage. Remember we said that the perichondrium is the outermost layer of cartilage? Well, you really won't find perichondrium on articular cartilage, and this is not very common. This is a unique area where you just won't find the perichondrium. We have the epiphysis of the bone, we have the spongy area of the bone, and we have the compact bone. Remember we said that the compact bone is the osteoid. It contains a lot of osteon or osteoid, and you have the cavity, which is the trabecular bone or the softer area of the bone. This is basically showing the osteon with aversion system. So you should expect osteoblast that would synthesize this. Here, the spongy or trabecular bone, you see more of your osteoclasts, and the osteoclasts have specific function. So the osteoblast is responsible for laying down the bone. So the osteoblast synthesizes collagen, it synthesizes the extracellular matrix, while the osteocyte is just there to maintain the bone. It's just there to support the bone. It's the mature form, doesn't really have as many functions as the osteoblast. Then the osteoclast is there to do the opposite of what the osteoblast does, which is bone resorption. So osteoblast is creating the foundation to lay down your collagen, lay down your fibers, lay down your glycose, aminoglycans, your proteoglycans, while osteoclasts are producing enzymes to break down the bone. If the osteoclasts are producing enzymes to break down the bone, what important organelle would you expect to be in an osteoclast? Take a wild guess. If you're smart, you get A+. Well, you're all smart.
Vesicles?
Huh?
Vesicles?
Yes. You have vesicles, but how about lysosome?
Oh, yes.
With hydrolytic enzymes, just break down stuff. So that you'll find that is a very important thing about the osteoclast. Osteoclast is there to break down the bone. Remember, like every cells in your body, regeneration has to take place. Not all cells, remember some neuronal cells cannot regenerate, but there has to be a natural turnover of cells throughout your body. So most tissues you look at, you'll find that there's regeneration and remodeling of cells, just like the epithelial cells that claws off. The gastrointestinal cells, they change each time. The bone cells also do the same. The osteoclast will create new bone composition while the osteoclast will break it down. So there has to be a balance between the osteoblast and the osteoclast. Here, we said that you have hyaline cartilage at the articular surface of the bone, hyaline cartilage at the articular surface of bone, and there are different types of cartilage, 3 types. You know them? Hyaline is 1, fibrocartilage is 2, the other one.
Is it articular?
Well, articular is a type of hyaline cartilage. Elastic. You need to know all 3 of them. This is looking at the extracellular organization of the cartilage. We already touched on the protein, the extracellular matrix, what we expect to find. Remember, we said that the chondrocytes will create these protein. They are going to have an abundance of rough endoplasmic reticulum within it to synthesize these protein, and naturally they'll be packaged in the Golgi and released by exercise. This is just showing the proteoglycan. We said that there's a core protein. You also have hyaluronic acid, the GAG, which is disaccharide structures that are extending from the proteoglycan. This is the more enhanced view of this proteoglycan. The core protein is the core protein, and you have different sulfated groups on proteoglycan: chondroitin sulfate, dermatan sulfate, heparan sulfate you can also find, and the disaccharide group glycose aminoglycan. The glycose aminoglycans are negatively charged with side chain that they repel one another and attract water, which is why you have a lot of water as well in the extracellular matrix. This is also looking at the structure of the cartilage. We talk about the 3 types of cartilage right here. We said hyaline cartilage, fibrocartilage, elastic cartilage. What would you suspect the difference would be between all 3? Think about elastic. What type of fibers would you expect?
Elastin.
Exactly, you expect elastin, and you expect collagen fibers in hyaline. You also expect collagen fibers in fibrocartilage.
Yes. Is it that they're more organized in the hyaline than the fibro?
Exactly, and the hyaline cartilage is the most common, so you find it in most places in your body, hyaline cartilage. Fibrocartilage, you find it a few places like pubic symphysis, intervertebral disc. We'll touch on that a bit. The elastic cartilage, you find it in 3 places with E: epiglottis, the ear, and another area with E. My brain seems not what to recall right now. The nose, E, it starts with E. So we have epiglottis, the external ear, and eustachian tube, that's in the ear. This is showing a histology of the hyaline cartilage above. If you can see, we have the chondrocyte. You realize that there is a difference between the chondrocyte at the periphery, and I can't say perichondrium because, as I said, in articular cartilage, this is one of the few areas that it will be devoid of perichondrium. But the chondrocytes tend to be a bit more, they tend to look a bit smaller, but as you come inwards, you realize that you have calcification here. So you have calcified cartilage within, and sometimes what they refer to these as chondrocytes in lacunae. It seems like an empty space. Or they call it empty space, and you'll see the same thing also in the bone marrow or for the bone. It's an empty space because it's usually water that is within that area, and I think when they stain it or through handling or processing, that area just appears like an empty space. So 3 types of cartilage, you already know them: hyaline, elastic, fibrocartilage. You just need to know where you find them. This is chondrocyte lacunae. Hyaline cartilage, you expect chondrocytes to be the cell and you expect the matrix. Fibrocartilage, chondrocytes to be the cell and you expect the collagen fibers. Elastic cartilage, chondrocyte to be the cell, again in lacunae, just an empty space, and elastic fibers. These are the cells. You see the nuclei, these are the cells. The thing is, you're not really seeing the collagen fibers so strongly because the collagen fibers tend to not readily trap the stain. It gives this glassy appearance, and you basically won't see much of it sometimes in histology morphology, like the typical collagen fibers that you expect. Hyaline cartilage, you find them on your rib, the sternum, areas where the bones articulate. So ribs, sternum, synovial joint, as you already know, and they're abundant in your respiratory system. The trachea, the larynx, the bronchi. Remember, all of those have cartilage. And I think you were looking at one, Daniel. You remember?
In this week or last week?
I think last week you were looking at a histology of the trachea, and you
and I saw the
chondrocytes. So yeah, that was hyaline cartilage with chondrocytes in lacunae. So you know that they find them in your respiratory tract. So when you guys come again, you can look at trachea, because we have a lot of trachea. I'm not sure if we have bronchi, but if you find me, you can look and you will see the chondrocytes in lacunae, and you will see the perichondrial region. And remember, the perichondrium is the outermost area that is then connective tissue. The elastic cartilage now you find in the epiglottis, auditory tube or eustachian tube. So you find it in the external ear, the epiglottis, and the eustachian tube or the auditory tube. It's pretty rare. You don't really find much of the elastic fibers anywhere else so much in your body. You know that you have elastic fibers, yes, in the blood vessels, the fibers themselves, but not the cartilage, right? They have the cartilage, which would contain the chondrocytes in lacunae and the elastic fibers. So like the auricle of your ear, you realize if you twist your ear, if you fold it over on itself, it just recoil on itself. That's the elastin that is within it. You don't really have much places that you have elastic cartilage. It provides support and it tolerates distortion. It's able to recoil. And in terms of the fibrous cartilage, you won't find them many places either, but you'll definitely find them in the intervertebral disc. Between these areas, they call them annulus fibrosus. You expect to have fibrocartilage. You can also find it at the pubic symphysis, that little area where you have your pubic bone. And these are the chondrocytes again in lacunae. What's common about all three are chondrocytes in lacunae, and they have their extracellular matrix with either their elastic fiber, collagen fibers. This is just histology showing hyaline cartilage. We have a perichondrium here. Remember, we said that all of them are perichondrium. You remember where you don't find the perichondrium?
In the hyaline cartilage. On the outermost layer?
Yeah. Remember, the perichondrium is the outermost layer. You know, you don't really always find it everywhere. So for example, there was this. There's no perichondrium here, right? This is the hyaline cartilage. You don't find it in the articular cartilage, for example, in your knee, even though it's hyaline, but you can find perichondrium in other areas where you have hyaline cartilage. So for example, in the trachea, you'll see the perichondrium. I'm sure you see this. Look, if you look at the epithelium, this is the area. Let me orientate you a bit. What does this look like to you? What kind of epithelium? What's this white spot?
Oh, those are mucous.
It looks like goblet cell. You see it white? Anytime you see it white out like that, goblet cell, you really can't tell if this is pseudostratified, right? But once you see cilia, look at the top. You see the cilia on the top?
Yes, ma'am.
Cilia on the top, goblet cell. You can't really define whether it's simple or stratified. Take the wild guess and say it's pseudostratified because you have cartilage right here. Trachea has cartilage, right? Hyaline cartilage. So this is more likely the trachea, and you already know the territorial matrix and the chondrocytes in lacunae, as well as the perichondrium. So hyaline cartilage, as I said, is the most abundant cartilage in your body. Before you were born, at some time before 5 weeks gestation, all your bones that were formed out were actually hyaline cartilage. They were all hyaline cartilage, and they went through a process called endochondral ossification a bit later in life, where there is mineralization of the bone, where the cells literally start secreting the extracellular matrix and the minerals to harden the bone. But remember that the end of the bone, where you have the epiphyseal plate, that area cannot undergo ossification. Why? That area is the area in which you need to grow from. So if that area you undergo ossification early, you basically are dwarf, literally. Hyaline cartilage is to provide support to the skeleton skeleton, and it's replaced by endochondral bone formation. And you'll eventually understand that there are two ways in which a bone can form: endochondral and intramembranous. Endochondral ossification, intramembranous ossification. And the basic difference is that endochondral bone formation just means that the bone was formed from hyaline cartilage. So what cells would you expect to be synthesized in the extracellular matrix? Chondrocytes, right? Yeah. While intramembranous ossification is different. You expect osteoblast to be synthesized in the extracellular matrix and mineralizing that bone. So at birth up to adolescence, you have hyaline cartilage. It's an integral part of your entire system. It's an integral component of the epiphyseal growth plate, which control growth and shape of long bone. So the epiphyseal plates are the important part, right? So this is the hyaline cartilage. This is showing an area where primary ossification occurs. So ossification just means the bone is being formed. It's converting from cartilage to bone. So initially, you will have primary ossification center, where you'll have the mineralization start taking place in this area in the center. Eventually, you'll find that here, where you have the epiphyseal growth plates, this is called the secondary ossification center. Here is not mineralized or not fully closed until you're probably 21 as a female, I think up to 25 as a male, because naturally, if this area undergo ossification, then you'll stop increasing length. So you won't grow any taller. So you can just imagine that a person who is vertically challenged have a problem with secondary ossification or premature secondary ossification. So here, just showing the epiphyseal blood vessel, the epiphyseal plate. So you already know that hyaline cartilage are found in the lines, the articular surface of your synovial joints, where you find your synovial joints here, right? In your knee. They act as shock absorber. So naturally, the cartilage could not be as hard as the bone, because the cartilage is basically a shock absorber for the bone, which makes sense why it would be semi-rigid. Damaged hyaline cartilage is unable to repair, and you already know this. Why?
It's not vascularized.
Exactly, very good. So here we have the perichondrium, chondroblast in lacunae, and nothing that you don't know. So the perichondrium is a dense connective tissue, which is essential for cartilage growth. And if you look at the perichondrium, you realize that the cells tend to be smaller, the chondroblasts. They're the immature version of the cell. And remember, sometimes you can have fibroblasts as well. The innermost area of the cartilage, you tend to find the chondrocyte in lacunae. They're just more mature form. They're not really producing extracellular matrix and contributing so much to the structural composition. So the chondrocytes are flattened near the perichondrium and are round in deeper region. Chondrocytes in hyaline cartilage are often arranged in pairs or groups of 4 to 6. So here you see them in pairs, and here you can take a wild guess by just looking at the arrangement of the cells. They tend to be flatter in the perichondrium or perichondrial region, tend to be wider and much larger in the center. You expect the chondroblasts here, chondrocytes here, chondrocytes in lacunae. Hyaline cartilage, I mentioned previously that the collagen fibers can be different when you're looking at cartilage versus bone. Cartilage, you tend to have type 2 collagen fibers, while bone, you tend to have type 1 collagen fibers. Type 2 collagen fibers, it has some water-bound substance, glycosaminoglycan, chondroitin sulfate. The proteoglycan, which is the bigger term, can be sulfated, and we already went through this. So in addition to the proteoglycan, the water, the fibers, we know that you can also have macromolecules. The macromolecules can include all of what we've mentioned before, just other protein, proteoglycans, and fibers. Those are basically macromolecules. This is just looking a little bit closer on the structure of the proteoglycan. The proteoglycans are composed of core protein with complex carbohydrate known as GAGs. You guys already well familiar with GAGs, and the GAGs look like bristle brush. So these are just the extension from the core protein. Their carbohydrates are disaccharide chain. As we mentioned before, GAG consists of repeating negatively charged disaccharides, and remember why we say the negative charge does, it attracts the water, and it consists of units of various length and may be sulfated or non-sulfated. So the proteoglycans can have, we already know, chondroitin sulfate, keratin sulfate. You'll also see heparan sulfate, hyaluronic acid, core protein. You can have sulfated groups on each. And this is basically just showing the same thing magnified. So the clinical point, you can have osteoarthritis. Osteoarthritis is a disease of the articular cartilage. This is basically when the extracellular matrix is degraded and there's altered chondrocyte metabolism. So you have a decreased glycosaminoglycans, there's increased water. Remember, if the glycosaminoglycan is decreased, well, you're going to have increase in the water content. In medicine, we call that joint effusion. It just means that there's more fluid in the joint. You also can have degradation of the matrix, breakdown of proteoglycans as well as the collagen. So it's basically osteoarthritis, and you'll know some old person in your life who will have osteoarthritis. Now you know, as the biomedical engineer, that you'll need to engineer all of these collagen that is missing, all of these proteoglycan structure that is missing. You will need to engineer all of this and the glycosaminoglycan that is necessary to prevent the joint effusion. So we already know you have three types of cartilage: elastic cartilage, fibrocartilage, hyaline cartilage. We already looked at the hyaline cartilage. The elastic cartilage contains chondrocytes and lacunae, nothing that we expect that it should be distinctly different from the others, except that you have elastin, right? Elastic fibers. You find them in the auricle of the ears, as we mentioned, the epiglottis, eustachian tube, or auditory tube. So here you expect to have a perichondrium, and you have the chondrocytes in lacunae, and you tend to have the elastic fibers highly stained. Here we have the elastic fibers between the chondrocytes in lacunae. The lacunae is just space here, and you expect the perichondrium. So elastic fiber is enveloped by perichondrium. Remember, we said that there are some cartilage, or probably just one or a few, that don't have the perichondrium. Fibrocartilage don't have perichondrium, but you tend to have it in hyaline cartilage and elastic cartilage. However, in hyaline cartilage, the articular cartilage in your knee, you tend to not have perichondrium. Cartilage is enveloped by perichondrium, and it undergoes appositional growth from the perichondrium, or interstitial growth by chondrocyte mitosis. So what this means is that appositional growth means that it expands in width. So when the chondrocytes are producing the extracellular matrix, it increases in width, while interstitial growth means that when the chondrocytes are laying down the collagen, the extracellular matrix, whatever it is, the elastin, it basically extends vertically. So one, it widens, and the other, interstitial, it lengthens. Elastic cartilage does not calcify with age, and that's a good thing. This is just showing histology of elastic cartilage. Nothing that you won't see readily. Perichondrium, this is the matrix, the chondrocyte in lacunae, nothing special about it. Perichondrium here, yeah. So fibrocartilage, no. Fibrocartilage, we already touched a bit on. You find it in intervertebral, this, talk about nucleus pulpus, and here, symphysis pubis, right here at your pubic bone, you tend to find it. Yeah, you find it like the meniscus. These are some cushion pads between your knee. You don't really need to know that, but if you do, that's good. And this is just a reminder that hyaline cartilage, you find the tracheal cartilage. You have different type of cartilage. I mean, you know the trachea, but you have cricoid cartilage and tracheal cartilage here. Bronchioles, bronchial cartilage, you tend to find the hyaline cartilage, and the elastic, we say you find it in the EEEs, and the fibrocartilage you find in the intervertebral disc, pubic symphysis. Just remember this long area come down right here, so that's where you find fibrocartilage. Fibrocartilage has great tensile strength. It attaches bone to tendon, and it's dense fibrous connective tissue that replace fibrocartilage. So remember that the cartilage tend to not reach regenerate. When this happens, fibrocartilage replaces a dense fibrous connective tissue. And we know you find it in annulus fibrosus of the intervertebral disc. That's just the other name for what you were looking at right here, the intervertebral disc, annulus fibrosus in the intervertebral disc, nucleus pulpus, annulus fibrosus in the intervertebral disc. This is a vertebral body, and remember you have different vertebral bodies going all the way up, and you have the cartilage in between that's there for cushion and for tensile strength. So you expect to find no perichondrium. I hope that's what this means. No, nucleus pulpus. Anyway, you expect to find no perichondrium in fibrocartilage. You expect to find your matrix again with the collagen, and you expect to find the chondrocytes in lacunae. So this is just showing the histology of the nucleus pulpus here, and this is just a bigger view of the diagram. Fibrocartilage, as I said, lacks perichondrium. The matrix has collagen fibers. You have your ground substance, and in contrast to hyaline cartilage, remember we said cartilage tend to have type 2 collagen while bone tend to have type 1. This is the outlier. Fibrocartilage can have type 1 collagen. Don't worry yourself too much as to what have type 1, type 2. It's just for you to know that there are differences, but I wouldn't ask you to remember, which one has type 1, which one has type 2. That's a bit much. It's good to know that that there's collagen. Understand a little bit about the structural composition of the collagen, like what it is, and that should be good. So expect a mixture of chondrocyte and fibroblast, and as I said, you can expect that sometime in cartilage. You can expect the chondrocytes and the fibroblast. If you look here, there's no perichondrium, right? No perichondrium, no dense connective tissue. In terms of the chondrocytes themselves, we know already that they synthesize and secrete all the components of the extracellular matrix. Similar to fibroblast, osteoblast, they synthesize the protein, they synthesize the collagen, the carbohydrate, and the ground substance. This is just looking a bit closer on the chondrocyte. There is the nucleus, you have the rough endoplasmic reticulum. As we say, you're going to have a lot of those because it's synthesizing a lot of proteins, a lot of fibers, a lot of structures. You expect a lot of free ribosome, rough endoplasmic reticulum. As I just mentioned, you expect to have Golgi complex. Remember that you're going to have packaging, and you're going to have vacuoles, saccules, and release by exocytosis. You expect mitochondria as well, and nothing special about that. So in terms of the formation of bone is formed when cells migrate. The cells migrate, there's mitosis, division, there's differentiation. For example, it differentiates from a progenitor cell, mesenchymal cells, to an osteoblast. That's the differentiation. The osteoblast will lay down the structure of the bone. There's modulation. It basically forms the structural model. There's the synthesis of a lot of things from these osteoblasts, and these osteoblasts will secrete the collagen, secrete stuff. There's also extracellular mineralization, and we already spoke a little bit on mineralization of the bone, and we already said that there's resorption. There has to be resorption. Remember what cells are responsible for bone resorption? Osteoclast. So here we have the growth plate, the epiphysis, and this is just showing immature chondrocyte. If you see an immature chondrocyte forming bone, what do you expect? I haven't gone through the types of ossification, but I mentioned it. Huh?
You're asking for the type of ossification?
Yeah. Remember, you have intramembranous endochondral. I would say that the hyaline cartilage
The endo one.
Endochondral ossification. Very good. And you have the primary ossification center, the secondary ossification center, hyaline cartilage. Primary ossification, secondary ossification. Remember, the primary ossification occurs from the center, the secondary ossification along the growth plate, where you have the epiphysis or epiphysis. Bone formation, you already know, can be intramembranous or endochondral. And it just means that the bone is derived from cartilage in endochondral ossification. And you know that most of your long bones in your body are derived from endochondral ossification, because in utero at around 4 weeks, most of your bones were hyaline cartilage before they became ossified into actual bone. Or can be intramembranous, where it's not a cartilage, but the bone is basically just formed from osteoblast. Intramembranous membranous ossification, bones that tend to be derived from intramembranous ossification tend to be the flat bone of your skull. So like the skull bones tend to be intramembranous ossification. And these are formed much earlier. And it makes sense. Biology, intelligence by design. When you're born, your skull's soft, but intramembranous ossification is more rapid than the endochondral ossification. This is just showing the growth plate, the epiphysis, the diaphysis. You'll eventually see that these are just structures with different types of cells that will determine how the bone grows or lengthens. The intramembranous ossification occurs in areas of ordinary mesenchyme, when ordinary mesenchyme differentiate into osteoblast. So it's not chondroblast, it's osteoblast in this case, right? Because it's intramembranous. Our bone forming cell differentiate directly within richly vascularized mesenchymal connective tissue. We already mentioned that the flat bones of the cranium and the mandible, well, we never said the mandible, but now you know the mandible as well. This is the bone in your jaw. Not sure why this stuff ossified so quick, but I guess it means you need to eat early. I don't know. But yeah, these bones are intramembranous ossification, where you tend to have the long bones like here, humerus, femur, sacrum. All of these bones tend to be from endochondral ossification. So these are all coming from cartilage. This is just showing a little bit more on the ossification itself. The mesenchymal cells are the progenitor cells that give rise to the osteoblast. Remember that the osteoblast, when they derive from the mesenchymal cells, they will start laying down the osteoid. Remember, we talked about the osteoid, and we said that the osteoid was just collagen, the organic fibers. Inorganic would be the mineralized or the hydroxyapatite. So when the bones start get hardened, so the osteoids will be produced by the osteoblast. This creates a primary ossification center. The osteoblast produce the collagen, create the osteoids around it. And of course, the osteoblast will eventually mature into osteocytes. But the osteoblast will also create calcification around it. So when the calcification occurs, that's when it hardens. So if it's only osteoblast and collagen fibers, it's soft. It needs to be ossified, it needs to be calcified, it needs to be hardened. When it gets hardened, the osteoblast will mature into osteocyte, and they'll just be trapped in this area, osteocytes in lacunae. It will just be trapped in this area because it's permanent, mature. It's not doing anything but supporting the bone marrow or supporting the bone. So when mesenchymal cells aggregate at site of richly vascularized connective tissue, they differentiate into osteoblast. So that's the difference. You need rich blood supply, you need oxygen in order for the ossification to take place. So they secrete osteoids, as you know, and it's an organic matrix of proteoglycans and type 1 collagen. Then the osteoblast also secrete alkaline phosphatase. What this alkaline phosphatase will do, it will cause the mineralization of the bone. And the most dominant mineral, calcium hydroxyapatite. It's a dominant mineral in the bone. This is what makes your bone tough. Hydroxyapatite. The osteoblast are going to just come out, synthesize the osteoid, synthesize the collagen, form the organic, and then the alkaline phosphatase, which will basically induce the mineralization, and the hydroxyapatite is the dominant mineral that will make the bone hard. So this is just a diagram of intramembranous ossification. We already touched on the mesenchymal cells that will differentiate into osteoblast. The osteoblast will secrete a little collagen from it by exocytosis, and these collagen fibers will form the organic matrix, which is the osteoid, and it will also form calcification or mineralization. And when it's mineralized, these cells become osteocytes, and the osteocytes are just going to be trapped in this area, lacunae, with a little space. And what you're seeing are another osteocyte. How the osteocytes actually communicate, they have extension. So you'll realize that the osteocytes tend to be a little bit different structurally from the osteoblasts because they have canaliculi. These are some external projections from the cell. So what these canaliculi are doing, they're communicating with the other osteocytes, using these canaliculi to communicate in terms of transmitting nutrients. And a matter of fact, you also have gap junctions in here as well. This is just the osteoblast. I think think I was trying to explain this in the first diagram earlier. The osteoblast creating the osteoid, and I said something about concentric lamellae and the aversion system. You have an aversion canal. This basically will contain the nerve, the blood vessels, and you have the osteocytes. You're going to have multiple osteocytes now surrounding this canaliculae, and they're just communicating with each other. So with intramembranous ossification, the osteoblasts are entrapped into the matrix. They become osteocytes. These spider-shaped cells reside in small spaces called lacunae, you already know, and they are connected with neighboring osteocytes by slender process that lie in small channel called canaliculi. Small islands or trabeculae of newly formed bones are initially laid down. So you're going to have small islands of bone that is formed, and you call that trabeculae. The trabeculae is the spongy part of the bone. Remember, we said the bone can be compact, this outer layer, which contains all the osteoids that are calcified. You can also have the trabeculae, which is the spongy part of the bone. And then you have large multinucleated cell osteoclasts migrate to the trabeculae surface to begin resorption of the bony matrix. This is just showing the osteon, the aversion system, the artery, the nerve, the vein. The artery, the nerve, the vein is just the aversion system within it. So it's just the blood supply, how the bone gets its blood supply. And we already mentioned the concentric lamellae of the matrix is basically the osteon. And what you'll have, remember, when the osteon is mineralized, it becomes hardened and the osteocytes are trapped in it. You're going to have multiple osteocytes in these concentric lamellae, extending their canaliculi, communicating with different osteocytes. And we already know the external layer of the bone is the periosteum, which is also a specialized connective tissue, just like the perichondrium. So endochondral bone formation, already know this, forms by hyaline cartilage. The first of two or more ossification centers appear in the shaft or the diaphysis. Remember, we're looking at the epiphysis, which is the top. The middle area is the diaphysis. So you have the primary ossification center that occur here in the diaphysis, and the secondary ossification center occur here in the epiphysis, and it's usually later in life. Here, the hyaline cartilage will convert to bone, or it will calcify and eventually will calcify. But this has to be later in life, because if it calcify early, remember, say, they are going to turn dwarf. Thin bony color appears around the diaphysis by intramembranous ossification as bone is laid down directly by a connective tissue perichondrium of cartilage. With everything that happens, I might not be telling you like the genetics, but there are growth factors, there are genetic signaling that are stimulating all of these things to happen. They're not just happening automatically. Remember that the cartilage on the outer area of the cartilage is perichondrium. In order for it to convert from a cartilage to a bone, it will have to convert to periosteum, right? Because the outermost layer of the bone is periosteum. So this is basically when in the primary ossification center, the diaphysis converts from perichondrium to periosteum. This is your endochondral ossification. In endochondral ossification, chondrocytes are arranged in columns, and they appear as two fronts on both sides of the central region. They eventually form the epiphyseal growth plate. These same chondrocytes will form the epiphyseal growth plate at the junction between the epiphysis and the diaphysis. So the growth plate up here, that's basically what they're making mention to. The growth plates of hyaline cartilage determine the longitudinal diaphysis. Remember, the tall you are is determined by the epiphysis. And these growth plates, the epiphyseal growth plates are closed after adolescence period. So when you stop grow, the growth hormone stops signal. So this is the end of your genetic expression. You're supposed to be 5 point whatever inch tall. That's basically when the growth plate will close, and it's just signal to your body that you stop grow. For example, like me, you obviously stop grow a long time, right? I think you guys too. You guys are big enough.
Fortunately.
Unfortunately. You're not that short though.
Yeah, but you know, 2 more inches could help.
Right, you'll be fine, I'm sure. Most long bones, the vertebral column, the ribs, the pelvis, develop by this way, as you already know. Mesenchymal cells differentiate into chondrocytes, and the cartilage template is modified to facilitate mineralization, vascular invasion, and replacement by bone. Both kinds of osteogenesis have the same mechanism of bone depositions. The deposition of the collagen, the fibers, the extracellular matrix is just going to be the same. It's just that the cells are different. One is chondrocyte, the other is what? What is the other? Osteoblasts, exactly. And this is just showing endochondral ossification, which you already know. At 8 weeks, you have the hyaline cartilage, the perichondrium, and you have hypertrophic calcified cartilage. You form an ossification center. By the time you're 10 weeks, it forms a periosteum. And of course, here you still have your cartilage, and the secondary ossification center here in the epiphysis. This is at 10 years. You still have articular cartilage. Well, you still have the epiphyseal growth plate, or it hasn't closed. So the clinical point, you have Paget's disease. I tried to take out some of the clinical points because you don't need to know all of them. It's just exciting if you do know them. The osteodystrophia deformans is a chronic disorder of excessive bone resorption. So it's like the osteoclasts are overactive, just resorbing the bone, breaking down the bone. This is a mutation as well. You guys probably can figure this out or help me figure it out. The etiology is unknown, but it's usually due to a viral infection. A lot of things are due to viruses. Remember, we said viruses are just so sinister. So this is a genetic mutation, and this is the gene that is responsible. It's easy to test for these genes or to see if there's mutation in these genes. But what will happen in these patients is that because the osteoclast is not functioning optimally, they have a lot of resorption of the bone, so the bone gets brittle. It's breaking down the bone more than it's building up the bone. The osteoblast can't build up the bone fast enough as the osteoclast is breaking down the bone, so you basically have misshapen bone, the bone becomes brittle, and I guess these people will be susceptible to pathological fractures. Growth plate in cartilaginous bone, we already know here is the growth plate. Zooming in on the growth plate, you realize that there are different zones and the different cells that are there with different functions. So for example, you have the zone of reserve, you have the proliferative zone where you just have rows of chondrocytes coming down. You have zone of hypertrophy. Hypertrophy just means that it get a little bit enlarged. You know, when you exercise and your muscles start getting, it's hypertrophy of the muscle cells. So hypertrophy just means that the cells are getting a little bit enlarged, and as you can see down here, you also have a zone of calcification, zone of ossification. This is just the histology showing the difference between them. So the plate promote appositional growth. You remember what appositional growth is? Growth of hyaline cartilage.
It is increasing width.
Yeah. So growth of hyaline cartilage at the end facing the epiphysis. So that's how you get tall. Cartilage is structured in the lower region and replacement with primary spongy bone in the deepest region of the metaphysis. So you have 5 histologically distinct transverse zones of the growth plate, and they reflect the sequence of events in endochondral ossification. The zone of reserve is when you have the resting cartilage. It consists of cells that are flat and round, randomly arranged chondrocytes. So this would be the zone of reserve here, just small chondrocytes. Then you have the zone of proliferation. This is where the chondrocytes undergo proliferation. All of these are induced by something, in this case growth hormone, and you basically have columns of cells. So this is the zone of proliferation. We have the columns of cells. And you have zone of maturation or hypertrophy, where the cells become a little bit larger and they're in lacunae, and you can see them down here. Then you have the zone of provisional calcification, where you have calcification of the cartilage, and this area obviously would be a bit harder. And then you have osteoblast that can differentiate from marrow cells. And then you have the ossification zone, and this ossification zone obviously now comes down into the metaphysis, and this is when you have specules or calcified cartilage in this area, and this area is where it becomes mineralized. This is just another histology showing the growth plate and the metaphysis. Nothing that I haven't mentioned before. The zone of reserve, proliferative zone. You guys can just look at it in your spare time when you get the lecture. So in terms of bone resorption, we already mentioned that the osteoblasts are responsible for producing the bone, making up the bone cells, producing the cells, making up the bone extracellular matrix, the fibers, all of that. The osteoclasts, they are some giant multinucleated cells. They have lysosome and other enzyme, hydrolytic enzyme that break down the bone. So they're responsible for bone resorption. Osteomyelitis is just an inflammation of the bone. It can be due to bacteria, virus, parasite, anything that invade the bone and cause inflammation of the bone. Nothing special to know there. The cells of trabecular bone include the osteoprogenitor cells. These are just the stem cell that will differentiate giving you osteoblast. You have the osteoblast, which you already know makes up the bone structure. The osteocytes, which are the mature ones that are just sitting in lacunae for support. And you have the osteoclasts, which are the giant multinucleated cells that are responsible for bone resorption. You already know the microarchitecture of the bone. We said that it can be compact, the outer ardent area, the periosteum area, which is connective tissue, and the spongy, which you expect to be in here, the trabecular region. So you see it here, spongy bone here, and the compact bone. And you have the osteon with the eversion system, which include the arteries, veins, nerves. This is how the bone gets its blood supply. A little bit more detail, it's just saying that the osteocytes in lacunae are arranged circumferentially around central canal. They have a little central canal here. So the eversion canal connects with other canals with the medullary and the marrow cavity. So these eversion canal, they're just connecting. Remember, you have the osteoid, and within it you have canal. They are communicating. So there are different canals communicating. And remember that you have the osteocytes that are also communicating via canaliculi, allowing diffusion, and with gap junction, allowing things to pass between one osteocyte to the next, from one osteocyte to the next. This is just showing the eversion canal and Volkmann canal. This is just how they communicate here. Volkmann canal with the blood vessel running through. Volkmann canal here. Eversion canal, the eversion canal, and this is how they communicate one canal to the next. Osteoarthritis, where you have osteophytes. This is basically when you have the bone growing out and sticking out. So for example, if you have this in between your knee and between the articular cartilage, just imagine you growing pieces of bone sticking out. Remember, the cartilage is there for cushion, and you have dysregulated bone growth, inflammation that is producing bone pieces or bone spur, and this will basically cause erosion naturally, right? So patients with osteoarthritis that goes through all of this wear and tear producing osteophytes. They have difficulty walking, difficulty just standing in general, and you have a lot of pain in that area. You already know the spongy bone, the compact bone, the cortical compact bone, as well as the spongy bone, the inner region of the bone. So the compact bone can form or resorb beneath the periosteum or on the endosteum. With aging, bone girth increases, but thickness and density decrease. Compact bone also remodels by forming osteon, which are all aligned in the same direction to resist bending force. The outer periosteum provides root for vessels and nerves, actively participate in bone growth, repair after fracture. So this is just a histology showing what looks like eversion canal. This should be the canaliculi. Well, no, you shouldn't see the canaliculi, but this is the osteon, and these, you're supposed to have the cells, osteocyte, and this is supposed to be the periosteum, or the outermost region of the bone. It's supposed to be the spongy region of the bone. So you have the compact bone out here. We have most of the osteoid or the eversion system, and you should have the spongy bone. This should be where you have the bone marrow, and yeah, that's the spongy bone and the compact bone. That's just what the histology is showing. Except where bone articulates with other bones in joints, the outer outer surface is covered by tough, fibrous, highly specialized connective tissue, and outer layer of dense, irregular connective tissue consists mostly of fibroblasts. You already know that the bone tends to have type 1 collagen as opposed to the cartilage. The periosteum contains large blood vessels, nerve, lymphatic, and contain osteogenetic cells, the osteoblast. And you already know the eversion canal, which supply or connect the Volkmann canal with the blood supply. In terms of the formation and composition of collagen, you don't need to know all the details, just have an appreciation that collagen can be synthesized by osteoblast, chondrocyte, fibroblast. It requires these cells to have a lot of rough endoplasmic reticulum, because the rough endoplasmic reticulum is where the synthesis will take place, will be packaged and released to the external or the extracellular matrix from these cells. You expect translation, hydroxylation of different amino acid, proline, lysine. If you like the detail, you can remember these. And glycosylation, forming pro-collagen, and eventually the triple helix formation. You get a pro-collagen, and it's just basically protein structures forming a triple helix being synthesized within the rough endoplasmic reticulum and secreted from the cell. This is just a step-by-step detail. Don't need to know it, but you can read it to understand it. This is showing the same thing, same proline, lysine, proline to the pro-collagen, rough endoplasmic reticulum. Eventually it forms hydroxyapatite crystals when you have mineralization. Osteogenesis imperfecta is a hereditary disorder that is characterized by fragility and deformity. They tend to have recurrent fractures. Why? Them have deficiency of type 1 collagen, or the collagen synthesis is impaired, and this affects the pathway, the collagen synthesis pathway. As you can see, you needed the alpha protein and the pro-alpha 2 chains. You have abnormalities here. So if you have a bone without collagen, remember it's a crucial component of the bone, then the bone becomes brittle. So it's called osteogenesis imperfecta. That's just giving you the perspective as to how many things can happen. You learning all of these things about cells, about proteins, fibers. That's basically medicine. So the cells of bone formation, and not going to take much longer, we're almost at the end. We already mentioned that you have the 4 types of cell. You have the osteoprogenitor cells. These are the cells that can divide. They have stem cell properties that can divide, give you your osteoblast to create the bone. Then you have the osteoblast that can make the bone, osteocytes, osteoclasts. Osteoblasts, which they synthesize and secrete osteoid, and they consist of type 1 collagen and non-collagen glycoproteins such as osteocalcin, osteopoietin. These cells also synthesize the alkaline phosphatase. Remember, we said that alkaline phosphatase, it will promote mineralization, and the hydroxyapatite is what will make the bone hard. So you know that the osteoblasts are going to have a lot of rough endoplasmic reticulum because it's synthesizing the osteoid, is synthesizing the collagen. This is just showing the collagen fibers on electron micrograph, showing the nucleus of the osteoblast, mitochondria, the extensive rough endoplasmic reticulum, the fibers, the collagen fibers on the external surface or the extracellular matrix. The osteoblasts, we know they have a lot of Golgi complex as well, because remember they have to be packaged and assorted and secreted by exocytosis. And there's gap junctions between these adjacent cells that plays a role in mineral metabolism. The osteoblasts, them have receptors. Remember that cells have cell membrane with receptors. They're going to interact with receptors from the parathyroid gland called the parathyroid hormone, estrogen, progesterone. If you learn a little bit more details about this, you'll realize that with the decrease in estrogen when you get older, that's why older females tend to have bone problem or osteoporosis because of the deficiency of estrogen. That's just the medical correlation. Osteocytes, now you already know that they're the mature version. They reside in the extracellular matrix in lacunae. Each cell display cytoplasmic process. Remember, we said that the cytoplasmic process are called canaliculi. See it here? It's on electron microscope or micrograph. External projection, and these are contacting with the other osteocyte, and they're using these canaliculi for communicating with the other osteocytes. So the process of one cell are linked to those of the adjacent cell by gap junction. Extracellular fluid in the canaliculi permits transfer of molecules, so oxygen, nutrients, stuff can diffuse across these canaliculi from one osteocyte to the next. So they exchange ions and other mineral with extracellular fluid, and this is a process they call Osteotic Osteolysis. Osteoclasts, we already know, are multinucleated giant cells. They can have up to 50 nuclei, so they are multinucleated. So if I ask you which one of them multinucleated, you know it's the osteoclasts, and you already know that the osteoclasts are responsible for bone resorption. So they break down the bone while the osteoblast synthesize the bone. The osteoclasts, they have a thing called, just like how the osteocytes are in lacunae, the osteoclasts are found in cavity known as house-shaped lacunae. If I show you a cell, or if I say the osteoclasts are within a cavity, or the osteocyte, for example, the osteocyte is within a cavity, you know it's lacunae. If the osteoclast is within a cavity, it's house-shaped lacunae. They have numerous mitochondria as expected, and we already mentioned that they have lysosome and membrane-bound vesicles, vacuoles. So you got it right, Daniel. You got the A+. Congrats. The vacuoles, the vesicles, small amount of rough endoplasmic reticulum, multiple Golgi complex. They are composed of extensive infolding at the border for resorption. So this infolding of the plasma membrane, they can increase the surface area and help to facilitate resorption. They also have cytoplasm rich in actin filament. The membrane of the border contains proton pump that keeps the pH low in resorption cavity. So imagine they're making it acidic for resorption. Secretion of hydrogen ion in the extracellular space dissolve inorganic material. So when they secrete the hydrogen ion, it dissolves the inorganic material in the matrix. And the lysosomes release hydrolytic enzymes, as we mentioned. These include collagenase, and you would suspect that the collagenase will basically just come and break down the collagen. So in terms of bone fractures and repair, successful regeneration requires a viable periosteum. Remember that the blood supply comes from the periosteum with effective apposition and immobilization. So you realize that when a person comes in with a fracture, you always have to immobilize it. So you wrap it up and it needs to be immobilized in order for the regeneration to occur. And that's a good thing about bone. You can break it up and it can grow back. You just need to put it back together properly and with effective apposition, because you can have callus being formed where the bone just forming fibrous tissues. Well, not necessarily fibrous tissue, but the bony structures are being formed, and for example, it would expand in width because it can contact the other end of the bone. So you basically have callus formation. So you have to ensure that the bone is approximated properly and properly immobilized in order for it to regenerate. So sometimes if you look here, you can add stuff to it to stabilize it. The repair of long bone, unlike flat bones of the skull skeleton, occur by intramembranous ossification and endochondral ossification. Healing will involve that you have a little inflammatory phase followed by a reparative phase and remodeling. What this means is that when you have the injury, you will have a blood clot form, an hematoma that is formed, then you have inflammatory response due to the death of the osteocytes. You have necrosis. You will basically now have infiltration of the fibroblasts and the new capillaries, new blood vessels in the periosteum, migration of a lot of cells to the area. That's basically inflammation. When this is inflammation, it just means you have a lot of signaling happening around the area where the injury happens. You have a lot of cells migrating to the area, leukocyte, monocytes, macrophage, and granulation tissues. These are just tissues that will help to regenerate the structure that is damaged. You also have osteoprogenitor cells from both periosteum and endosteum. And as I said, intermediate and late stage of those. So you know that the early stage is basically when you have the inflammatory cell, the hematoma, and a lot of cells run into the area to produce whatever it needs to stabilize it. The intermediate to late stage is when you can form callus. So this is what the stages are like. So So you have hematoma formation, fibrocartilaginous callus formation, bony callus formation, and remodeling. So you have to have a callus, but the thing is, sometimes you see a person get fractured and they don't put it properly together, not close apposition and adequate immobilization, then you have disfigured limb. In terms of engineering, bone tissue engineering, you can read this in your spare time. It's basically using stem cell technology. So you can use stem cells to recreate osteoblasts, which will basically help with regeneration of the bone. And there's also a signaling factor called bone morphogenic protein. So if you want to look into any biomedical, anything about engineering, it will always involve, well, most often stem cells are signaling factors and proteins, nanoparticles, maybe. And that's where the science is going. This is the last two. I added it because I had it before. Just synovial joint, synovial cavity. It contains confining sheet of tissue, the synovium, that produce synovial fluid. You already know that the articular cartilage are there too, and it's basically transmit the weight. You can also have a capsule with irregular connective tissue as well, and you know that it's hyaline cartilage. And that's the end. No question? No problem. All right.
No questions.
Okay, great. All right. So the other few lectures shouldn't be long. I'll try to keep them short. The last two were the most intense. So hopefully we can make it a little bit easier from now on, and you'll probably can have fun at the end. I think I saw the timetable today, and the tentative date for the exam is April 29. That's what I saw.
Okay. All right.
Yeah. All right. So see you next week.
Next week, miss. Oh, you said that so next week, what is the topic for next week?
So next week will be muscle tissue.
Muscle tissue mechanics?
Mm-hmm.
Muscle tissue mechanics? Yeah,
in terms of the lecture topic?
Yes.
Yeah, it should be the bone marrow, hemopoietic stem cell, or just the bone itself, or the cells, the marrow progenitor cells. So it will look more like on the cells that you find in your bloodstream. Most of them are produced from the bone marrow. So like the macrophages, lymphocytes, stuff that you already covered. It won't be too long. And that's it.
Then after we're supposed to do some things like mechanics of those tissues.
Yeah, something from mechanics. And I'm thinking probably just to cover the action potential in terms of nerve, muscle. Not too detailed, just cardiac action potential, cardiac muscle action potential, skeletal muscle, and nerve tissue. Yeah, that should be having a revision after that, and that should be it.
Okay, sounds good.
Yeah, all right.
Thank you. Have a good evening.
Thank you.